thank you for joining the part 2 of the workshop. As maybe some of you did not participate in the part 1, I will just shortly remind you about where we are located. So in the winter, we enjoy the beautiful northern lights because we are located quite far up north. And in the summers, we have constant days. So summer night looks like this, and the winter day is a little bit dark. We are located 1 hour flight from Sweden's capital, Stockholm, established in 1985, as you heard. And here you can see our 2 locations: antibody production facility based in outside Umeå, in this beautiful landscape of northern Sweden, and our laboratory located in Umeå, where we're doing antibody purification, quality control, and Western blots. We are ISO certified, and we are part of Olink Proteomics since the year 2020. And Olink Proteomics is a company which is developing kits for platforms to test the blood drop for the presence of thousands of proteins. So in one blood drop, one can check presence and quantity of different human proteins. And Agrisera is providing them with the antibodies for that test. Our products contain successful protein detection reagents. So antibodies for plant and algal cell biology is our focus, but we also provide you with epitope tag antibodies, high titer secondary antibodies, and detection reagents for both Western blot as well as ELISA. And now we are coming to the focus of our meeting. Here I would like to remind you the flow of the Western blot. So in part 1, I covered the steps from sample collection until protein transfer. And this part is available as a recording, which you will receive a link to if you would like to download it. I will share the slides also as a PDF with links, and there you will find the link to the recording of part 1. And today we are focusing on further steps of Western blot procedure, which hopefully are leading to the result as you see on this slide. And just to remind you how the visualization of a protein and a complex protein mix is done in a Western blot. It is done by an antibody, which looks like this, a Y-shaped molecule, and antibodies are binding to so-called epitopes. And here in the Western blot, we have a linear epitope, which is mainly detected. And as the name suggests, it is a subsequent linear epitope, are subsequent amino acids in the linear structure of the protein. And this is these are the epitopes to which antibodies are binding in techniques where proteins are denatured and unfolded. So mainly Western blot and, for example, immunohistochemistry. This is very important point for your understanding, and therefore I am repeating it from the meeting, from the lecture which we had in April. Then we have also a discontinuous epitopes. And as you see here, these are created by distant amino acids, which are brought together in the protein fold. And these epitopes are mainly recognized in techniques where proteins are in a native state, which can be immunoprecipitation, but also Western blot in native conditions, and chromatin immunoprecipitation. And the epitope as you see here, can be 2, but up to 15 amino acids long. Also, a very important point of your successful Western blot, which is very often overlooked, and therefore I am reminding it again, it is that antibody choice must match the species and the application. And I have met in my troubleshooting over the last 20 years very many cases where antibody was used, the wrong antibody was used on a target protein. What do I mean by that? Without checking conservation of the antigen used to develop the antibodies, researchers were assuming that it works in the pine, therefore it must work in Arabidopsis. It doesn't work that way. So if the species which you're working with is not listed in product information sheet from the supplier, then you need to ask if this antibody has predicted reactivity to the species you're working with. This is really crucial because if the short peptide used to elicit the antibody is not found in protein you're working with, your antibody will not give you a signal in a Western blot or in another technique. What do I mean that antibody must match application? One can predict to a certain degree if an antibody may work, for example, in chromatin immunoprecipitation or in some other native technique, because if an antigen is derived from the part of the protein which is hidden when protein is folded, the antibody is not going to work in a native technique. So without even going to the lab, you can save your time and determine if the antibody you would like to use is going to be suitable for protein and technique you're aiming to use it in. And please remember that one antibody, one validation. It means that antibody which works in Western blot may not work in immunohistochemistry. It is always a need to validate an antibody for a specific application to check if it performs correctly with a set of controls. As you see here in this presentation, there are also links where after you receive it, you can do the further reading, as the time frame does not allow me to go into all details. So how do I choose the right antibody for my protein? As you see here on the example of this protein of photosystem 2, the antibodies were made to 3 different parts of the protein. N-terminus, after a signal peptide was cleaved, inside of the protein, and C-terminal. So what I want to stress here is that that sequence to which the antibody is made needs to be found at least in 70% linear identity in the protein you're working with. Otherwise, the antibody is not going to work at all or very poorly. You may ask what happens if an antibody is made to a whole protein. Then there needs to be done an alignment, and also the rule is the same, that over 70% of linear sequence needs to be identical. So please, before you use any antibody, make sure that the peptide to which it is made is actually found in a protein you aim to detect. This is always how I start troubleshooting. I ask the customer on which species they used the antibody, and then I checked if there is any sequence conservation which will allow an antibody to bind. And now we're jumping into the procedure. So first, next step after gel electrophoresis and transfer of your protein to the membrane, using the transfer, which is wet or semi-dry, you have been having separating a complex mix on the membrane and transferring it first on a gel and then transferring it to the membrane. And why do we do the blocking? We do the blocking to, as the name suggests, make it impossible for antibody to bind to other places on the membrane than proteins. So all three places on the membrane need to be somehow made inaccessible. And in this example, blocking is made with milk. And please do not use these kind of tweezers because you can damage the membrane. The membrane has to be processed with big care and also wearing gloves. And is blocking temperature sensitive? Should it be done at 4 degrees room temperature or some other temperature? This is one of the questions I have received. So it is dependent upon protein antibody pair, which I will show you in the next example. As you noted here in this yellow text, if you use PVDF membrane, it can be dried after blocking and used 1 to 2 weeks later after reactivation. So PVDF membrane is much more robust and allows for this kind of procedure. And also we are storing our PVDF membranes for up to 6 months after they have been dried. And then we are using them later within 6 months. So one can prepare the membranes in advance. If you have some precious set of samples, they will be actually pretty safe when they will be stored on a membrane, as you can see here. And also drying up the membrane will additionally unfold the protein, because even in Western blot in the natured conditions, proteins may partially refold. That's why in some protocols, urea is included to loading buffer and to the gel to keep proteins unfolded and allow antibody binding. And drying the membrane will do just that. It will additionally unfold and fix the protein to the membrane. So you can actually include this step in your protocol if you're using PVDF membrane. And now we are coming to the background issue. This is the question I have received, that customer used a purified antibody, which was very weakly recognizing target protein. There were other bands coming up, and is it possible to clean up this primary antibody solution? And the answer is yes. And this is so-called depletion to clean up the primary antibody solution to remove these cross-reacting bands. So what we do is that we run gel electrophoresis, we transfer and transfer, but we are using the mutant cell extract, so lacking the target protein, so knockout. And we are incubating primary antibody solution with such membrane strip. So all contaminating antibodies will bind to contaminations, but as such a blot does not have target protein, the antibodies to target protein will remain in the solution, which you will use later. So this step will help to clean up the primary antibody solution. And also if you have some issues with, for example, Rubisco antibodies, they can be removed from your primary antibody solution in that way. And how do we block our membrane before we incubate with primary antibodies? With plant samples, it is mainly 5 to 10% non-fat milk, which is, as I see from the blots which I receive, the most efficient blocker. We also have commercial blockers, which are working well. If you need non-protein blocker, then it will be PVP40 used for in case of organelle fractionation. BSA is rather weak for certain type of samples, Non-fat milk is a mixture of different proteins, while BSA is just one single protein. So non-fat milk may more efficiently block a membrane. We also have a gelatin. And now let's look what result we can get. So as you see in this example, when the lab returned the first blot, they said the antibody does not really work. Look how many background bands, unspecific cross reactions. And then, as you see here, the blocking was done for only 30 minutes with 1% milk. I asked them to extend the blocking for up to 1 hour in 5% milk. And here you can see example how it worked. So more unspecific binding places were blocked and the better signal-to-noise ratio was obtained. And this blocking was done in room temperature. But in some cases, we may require to block overnight, as in this example. So blocking has to be adjusted to the specific protein-antibody pair, because sometimes it is enough to block at room temperature, but in some cases, overnight blocking is required. And I would like to point out, as you see here in a note, that if you're using avidin, streptavidin detection, you should not use milk as a blocker because there will be an interference. I mentioned best signal-to-noise ratio and how is it connected to the background. As you see here, in this example, in protein load, 4.5 microgram of total protein per well, and then primary antibody was quite concentrated. So, of course, the background bands were coming up. Diluting primary antibody is, of course, diluting also the background. And in this example, with a higher protein load, we will have more background coming up. This kind of optimization can be done once you know how your protein antibody pair is performing. So this is like refining the results and doing that titration. Background problem is, I think, one of the biggest problems in Western blot. So here I got a question, how to reduce the background signals while stripping and reprobing. And my question is, why can the background signal increase? Because we have a target protein on the membrane, and then when we are stripping, then we are also stripping we want to release the primary antibody from the target, but we may also wash away the target from the membrane. So when we are stripping, then especially for proteins of low abundance, we will decrease amount of a target protein on the membrane. Therefore, the signal-to-noise ratio will be changed, and the signal may be weaker, and therefore the background become higher. Many cases, I see attempts to compare different antibodies with each other. And therefore, I'm bringing up this point that in this experiment, researcher was comparing not fully characterized primary antibody with actin. And actin is high abundance protein, used often as a loading control. And it cannot be compared to a protein of low expression, which may require longer time to be visualized. So in general, I would treat each protein antibody pair separately and the way they are performing, because it depends upon biochemical properties of your target protein to which antibody is going to bind. And different antibodies are binding to different epitopes, as you saw. And to mention loading controls, which you're using, they are quite universal for both plant and mammalian samples. Of course, Rubisco is specific for plant samples, but otherwise histones, actin, and tubulin may be used as loading controls. However, I would like to remind you that the loading control cannot be determined a priori, cannot be determined without conducting the experiments, because in certain conditions, actin levels may change, like for example during stress. Therefore, it will not be a good control. Quite many proteins in the cells will change their expression profiles. So to be certain that you're using a proper loading control, one actually should do an mRNA profiling to check which proteins are actually constant. And now we are starting to incubate our primary antibody on a membrane. And you're probably using one of these buffers. As I see from troubleshootings, most of the labs are using TBST buffer. Minority is using PBST. But in this specific example, as you see, the background was lower when TBST was exchanged to PBST. That's quite interesting, and I unfortunately do not have an explanation for it. I would like to remind you that PBS buffer is not suitable when you're working with phosphospecific antibodies. Now we are starting the incubations with the primary antibody. And here I would like to show you the ways to save your time. Your time in the lab is really precious, and maybe you're dealing with some samples where you do not have too much of material. So you can actually incubate 3 antibodies on the same blot in parallel. So you can add them together. But of course, I would not recommend to do it for unknown antibodies. You need to know your antibodies and the dilutions, and you can mix them together and use on the same membrane. Of course, it is also possible to cut the membrane and, for example, incubate them one part with your antibody to your target protein and the other part, for example, to actin. So this is one of the ways to speed time, and it is described in more details in this blog post. Incubate 3 primary antibodies at the same time. A second method to save time is the following. We incubate everything at once. So primary, secondary, and blocking is done in one incubation. And this works for antibodies with high performance to proteins of high abundance. So this way you can shorten your Western blot protocol considerably. So you simply mix a blocker, primary, and secondary in one incubation. And then we have a third method. And of course, in this approach, we have no washes. We just develop the reaction after the incubation. So 3 in 1. And the third method where you can shorten your Western blot protocol is when we visualize target protein using directly conjugated primary antibody. In this example, it is an antibody to Rubisco, which is directly conjugated to horseradish peroxidase. It can be another enzyme or fluorophore. And this will also shorten your reaction time. However, I wouldn't recommend this method for proteins of low abundance, because for those, enhancement of a signal, which is coming from secondary antibody, will be quite desired. And talking about the secondary antibodies, just a reminder, they look like primary antibodies. They are immunoglobulins, which are making it possible for us to visualize the binding between primary antibody and a target. So antigen binding domain, and then we have FAB region and FC region. When you're choosing the secondary antibodies, you may sometimes see description that they are made to either the handle, so-called handle, FC region, or to FAB region where antigen binding domain is located. And we have secondary antibodies can be done in all those different species, and they are usually marked as H and L, which means heavy and light chains of a given immunoglobulin. And here a reminder, we have a target on the membrane. We use primary antibody, which binds to the target. Of course, the membrane is blocked, as you saw in a few slides ago. And then we have primary antibody made in the rabbit, in the host, which is rabbit. And then the secondary antibody has to be anti-rabbit. If you do not have signal in Western blot, it can be sometimes a reason that a wrong secondary antibody has been chosen. So you always need to have immunoglobulin in which primary antibody is developed. And if there is actually a difference between different secondary antibodies. So one of Agrisera secondary antibodies, called anti-rabbit, can be used in very high dilutions, 1 to 25,000. And as you see here, that antibody is quite efficient. It is also an antibody which is in a list of most published 10,000 most published antibodies in the world. And this antibody is extremely efficient. So why is it important to use to even bother about which secondary antibody you are using? The higher dilution you can apply of secondary antibody, you minimize the background signal. And incubation time, please never exceed 1 hour at room temperature. If you incubate too much of secondary antibody, it may actually lead to increased background signal. So 1 hour is definitely enough. And Agrisera secondary antibody, goat anti-rabbit, can be used, as I said, in dilution 1 to 25,000. So this will last for quite many more blots, maybe for whole your thesis. And it is desired to use secondary antibodies which are well performing, because it is also part of your success in the Western blot. And now think which secondary antibody you would choose for your Western blot. On a plant sample, for example, we have rabbit, goat anti-rabbit, horseradish peroxidase conjugated, and here we have goat anti-rabbit, horseradish conjugated with minimum cross-reactivity to IgG from these different species. So think for yourself, the first one or the second one, and I will show you the answer now. The first one is enough. And why is that? Because when we work with, for example, plants, we do not have in a sample immunoglobulins from these species. So using this, as it's called, adsorbed secondary antibody is not necessary here. Such antibody would be suitable for immunohistochemistry for visualization of protein coming from these species, as this would minimize potential cross-reactions. So in general, so-called adsorbed antibodies are more desired in other techniques, like, for example, immunohistochemistry, where we need to decrease the background. And now we are coming to detection, which can be done in different ways. Now we need to visualize our primary antibody binding to the target. And my favorite detection method is chemiluminescent detection. It is still one of the most popular detection methods. And here you see our ECL set, which is also included in the prize if you participate in the quiz. In chemiluminescent detection, we need to use the secondary antibody or primary antibody conjugated with enzyme horseradish peroxidase, and we need the CCD camera to visualize the detection. And here there is an example. It's often that too sensitive ECL is used, and therefore the background signal is increasing. So here in this example, Agrisera Super Bright, which is a extremely sensitive reagent, which has been used on a very abundant protein. So of course, if you wash a membrane, the same membrane, and develop it with less sensitive ECL, the background signal is minimized. So actually, you should have ECL reagents of two different sensitivities in your lab, and then you can save your blot. Because if you have too high background, you can just wash the membrane and then develop it with less sensitive ECL, as shown in this example. So your experiment is going to be saved. And of course, ECL has good advantages of sensitivity and quantification. Disadvantage is that recording of a signal needs to be done immediately. And it is appropriate for quantification and analysis of proteins present in low levels in the tissue. These are the advantages of ECL. And also with advantages, it comes also problems. You maybe seen these types of blots done with chemiluminescent detection. And here I will walk you through what can be a reason for this background. So as I mentioned a while ago, too concentrated but also too diluted secondary antibodies. So please read product information sheets before you choose the dilution of your primary or secondary antibody. Interference with plastic used to wrap a blot is actually very common because that can give you the interference and the background signal. Insufficient blocking, as you saw in the former slide. Insufficient washes also. So please stick to your protocol and apply washes with volumes and time as in the protocol. Poor membrane handling can also contribute to background issues. So you need to use gloves, forceps with flat ends, and keep always protein side should be up. And how do you know that the proteins, where is the protein side? You know it by cutting off the corner of a membrane after taking the membrane from the sandwich. And also, of course, contaminated buffers can also contribute to the background. So as you see here, you need to work according to GLP, good laboratory practices. So clean and with a good standard for your reagents and equipment. And also, increased background can be due to drying of a membrane during procedure. So all these parameters can influence the outcome. And as you see here, it is not the antibody here, but other factors which may influence that your blood does not look too good. And also, what is troublesome in Western blot, these are the blots from just last week, which I received. It is non-specific bands, which can appear. This is quite a common problem. So, primary antibody is recognizing epitopes on other proteins, and therefore, other bands are coming up. Target protein may be degraded or aggregated. So these especially if it's made to antibodies made to a whole protein, then we have much bigger epitope pool, which antibody is binding to, and therefore you can have such a ladder pattern. Actually, SDS may also interfere with proteins and be a reason for this phenomenon. And also, if there is no target protein on the membrane to allow specific antibody binding, then all other background bands will come up. In Western blot, we are after very good signal-to-noise ratio, which means that antibody is specifically binding to a target protein in amounts which allow for fast visualization of these binding. You know that if you expose your membranes for longer time, then all other bands are going to come up. If you expose membrane for 1 hour, it will look like this. What we are after is as short exposure time as possible, which will allow us to visualize binding of our antibody to specific target protein. And sometimes you maybe saw ghost bands. These are these white bands as on this blot, and you maybe wonder what's the reason for it. And actually, it is too much of everything. So you have too much of the protein, too concentrated primary, secondary antibody, and the reaction goes too fast to produce a black band. So then instead you will see a ghost band. A remedy for it is, of course, to decrease protein load, to use more diluted primary and secondary antibody. Chemiluminescent detection, as I mentioned, one can save the blot. If something went wrong, this was the question, if the blot does not develop properly or something went wrong during documentation, can the ECL incubation be redone? And you already know the answer for this question, and it is yes. And you can do a wash of such membrane, and you do not to strip, you apply another ECL of lower sensitivity, and then hopefully bands are visualized correctly. Next, second type of detection, which is also very commonly used, is fluorescence. It allows multiplexing, as you see in this example, so we can visualize two different proteins on the same blot. And we are using fluorescent dye conjugated secondary antibodies, and we need CCD camera for visualization. The advantage here, which is quite different from ECL detection, is that the signal will last for several months, and you can also multiplex several antibodies. But dedicated equipment is required, and the sensitivity may be lower than in case of ECL. Actually, ideally, if you work with proteins which are difficult, ideally it would be good to have two different possibilities for visualization in the lab. So have possibility to perform ECL and also to perform fluorescent detection. Because sometimes if one does not work well, the other actually can produce desired result. Fluorescent detection is suitable for quantification and multiplexing, as I mentioned. And then we have also a third detection method, which is a chromogenic detection method, where we do not need any CCD camera. We are using secondary antibody conjugated to so-called alkaline phosphatase, and we can visualize the target protein, and it is an inexpensive and fast method. However, please keep in mind that sensitivity is low. So this method will be good for proteins which are very abundant in the tissue. It will allow you a quick assessment, and then you can store the membrane strip in your lab book. You do not require any CCD camera for visualization of binding, but this is a method for proteins of high and medium abundance. I would not recommend this detection method for working with, for example, transcription factors. which are usually quite not abundant in big quantities. In my first lecture, I showed you in which databases you can actually check how much of your target protein may be in a tissue. And this is a guidance for which detection method to choose in the future. And when you are using this detection method, you should not use PBS for incubations because of the interference. It should always be TBS with this colorimetric detection method. So we have three methods which hopefully will provide us with good results. So we have chemiluminescence, fluorescence, and chromogenic detection method. Each of them have its advantages and disadvantages. It depends what proteins you are analyzing, to which method will be chosen. These two methods, chemi and fluorescent, are good for proteins of low abundancy, while chromogenic detection method is suitable when you're working with proteins of moderate and high abundancy in a tissue. How to determine molecular weight of a target protein detected in the Western blot? Of course, we're using the markers, colored markers for gel electrophoresis, which in our case we are combining with so-called magic markers, which will allow us visualization during Western blot development. Why am I bringing this up here? Is because there were cases where results were misinterpreted because there were no markers and the membrane was put upside down, and then result interpretation was completely wrong. So these markers, magic marks, are having IgG binding sites, and then they will be visualized when you are developing your reaction, and they are compatible with all three detection systems. So this is a really good and safe way to assess if you have been detecting protein of molecular weight, which is compatible with your target. You're probably using epitope tag antibodies. That was also one of the questions, and epitope tag antibody was not providing any signal. And it worked on one protein, which was GFP tagged, but not on the other protein, which was GFP tagged. And then there can be different reasons for that. And epitope tag antibodies sometimes fail to detect all tagged proteins which we are using in the lab. And what you can try, you first need to consider if expression level of your tagged protein was high enough, because one of the antibodies have a certain detection limit. If it's too low level of your expressed protein, that may lead to lack of detection. You can, in such case, increase protein load per well. You can also include the naturant 6 to 8 molar urea in extraction loading buffer. And what this denaturant will do is that it will unfold your target protein and allow the antibody to bind. For PVDF membrane, as you heard in previous slides, you can also dry it in the air, protein side up, which will be fixing proteins to membrane and further unfold them. If above doesn't work, you need to change to another GFP antibody. And you also always have to consider if your antibodies were properly stored, because that can also lead to their inactivation. Some antibodies may have a certain shelf life, which should be also specified on the product information sheet. So before you start any antibody work, you actually have to do certain preparation at your desk. and this will determine your success later. So read carefully product information sheets because the instructions should be there. If not, ask the provider. And when it comes to detection, I often get this question: how to get an intense signal from Western blot using a weak antibody? Well, first you have to go to the actually theoretical analysis and ask yourself if you used a proper organ, tissue, if it was in a need of fractionation, if your target protein was of low abundancy. For example, transcription factors are very rarely visualized in a total cell extract. Very often their levels are so low that fractionation is necessary to concentrate your target protein and facilitate the visualization using the antibody. It's simply concentrated to detectable levels by fractionation. You also maybe need to increase protein load per well. Did you have optimal transfer conditions? In my first lecture, I was showing you what is the difference between transfer conditions for proteins of low molecular weight and high molecular weight, because there is no one protocol fits them all. There's no such thing. You always have to consider biochemical properties of your protein, its molecular weight, and adjust the protocol accordingly. Also, antigen purified antibodies are of advantage for different techniques, and you have to use appropriate detection reagent. So as I mentioned previously, chromogenic detection is not suitable for transcription factors. So all these steps are called optimization. So if you do not receive a signal from your antibody at all, you need to, instead of doing repeatedly more and more blots, you need to do this theoretical analysis first. Here it's an example of optimization. This lab has been using the conditions as you see here. 20 microgram of protein was loaded. Blocking was done with 5% milk, and primary antibody was used overnight. Unspecific bands, right? The antibody is not really that good, or the protocol wasn't good enough. As you see here, the only change was using 10 times less of protein load per well. So this has to be optimized for each protein antibody pair. You cannot use one protocol for all protein antibody pairs. As you see here, this antibody was quite strong, so lowering protein load per well was eliminating the background bands. I have an advice for beginners: record a correct band first before multiplying variations of conditions, because this is what is often happening. There is blot after blot being done instead of doing the theoretical analysis of conditions and of the protocol. So if you are working with antibody which is not characterized yet, this is what we do. We do high protein load per well. We also block at first on the 1 hour room temperature with 5% non-fat milk. 1% is usually too low. And then, as you see here, we use primary antibody very concentrated for a long incubation. And of course, this can create a lot of background, but this is oversaturating the system. So our goal here is to obtain a signal of specific for specific antibody. And if you're using unknown antibody, you always have to include a control. In this case, it is a knockout mutant which lacks the protein. It is not expressed in that mutant. Therefore, on a Western blot, there should be no band in the mutant. It is very easy to obtain false positive results with antibodies. So if you work with uncharacterized antibodies, you also always need to include specific controls. I have also an advice for experienced users. Uh, One protocol does not fit all antibodies. Please consider that. I know that you have done a lot of blots, but still, with each new antibody, you need to reassess your protocol and consider if maybe transfer conditions should be changed, or maybe perhaps you should use fractionation because the protein cannot be detected in a total cell extract. I talked about it in my first presentation, which we had in April. So if you still have such blots, you're very welcome to contact Agrisera, and I will be happy to help you. And now it's time for a little questions. And first, I have questions to you before you can ask me questions. Uh, is it true or false? Other antibodies I have bought from Agrisera work. Therefore, this one should work too. Is this true or false? Answer yourself, and I will provide you with the answer. It's false. Each protein antibody pair is unique, so you cannot assume that one antibody purchased from the company is working, then the other one is going to work too. This does not work like that. Each protein antibody pair is unique. The antibody works for species A, so for sure it's also going to work for species B. As you saw, antibodies are made to specific peptides, and they need to be found in a protein you are analyzing. So this is false. One should always check the sequence to which antibody was made if it's also found in a protein which we are analyzing. The same protocol can be applied for all antibodies. The answer is false or true? False. Of course not. Antibody gives a good signal in Western blot, so therefore it should also work in other techniques. If you remember, it's false because one technique, one validation. Antibody used in Western blot is validated only for Western blot. It may not work in other techniques, and it requires a validation for that specific technique with controls. And I would like to bring up this important topic of how do we cite the antibodies. Please keep in mind to always include antibody name, product number, and supplier. And this way, you will help yourself and your colleagues and increase reproducibility. Because if a supplier has 30 antibodies to GFP and you do not include product number, then your colleague will not be able to reproduce your experiments. So it is very scary, but more than a half of antibodies used in research cannot be identified because in publications they are lacking product number and supplier. And on our technical blog, you will find posts where I'm describing different issues with antibodies and Western blot and what to do. And I'm going to be also active on the forum which Yao Hong created. If you're a plant scientist, you can benefit from our free educational posters about photosynthesis and epigenetics. You can request them. And also you can check which conferences are happening worldwide. We are running the Global Plant Events calendar together with American Society of Plant Biology for the 5th year. And it's not only conferences, but also workshops and online events, which are free of charge. And you can also download free images for your publications and presentations. If you would like to receive news from us, you are welcome to sign up for our newsletter. And we also have this offer of over 100 free antibodies. And there are new antibodies added to that list each week. There's no limit of provided antibodies, and you can check which antibodies we are offering that way. And this is an exchange. We provide 50 microgram of purified antibody, and we would like to receive results in 3 months. And you're welcome to contact Yao Hong with any questions you may have about our products. Good luck with your blots, and you're always welcome to contact us with any questions you may have. And also, I would like to open questions and answer sessions.
Does anybody have any questions? Okay, there's questions from Miha here. Let's see. Okay, hello, Professor. Thank you for the presentation. I would like to ask, according to you, using stripping buffer is not a good way?
Well, it may work for proteins of high abundance, but if you're working with proteins which are of low abundance in the tissue, you may have issues with detection later. So I know it works for certain protein antibody pairs, but not always. So if I can, I rather avoid stripping.
Okay, thank you, Dr. Joanna. And the QR code to the quiz is in the chat box. Feel free to check it out. Does anybody else have any further questions? Please just write it in the chat box. Okay, another question from Cancy. Hello, Dr. Joanna. I'm curious about antibody purification using knockout mutant. Can you please elaborate on the procedure, including storage conditions? Thank you.
Yeah, it's in case your primary antibody is producing a lot of background and very weakly recognizing your target, you can do this type of cleaning of the primary antibody solution. So you prepare the membrane with several lines of your mutant extract, which is from a knockout mutant, and then you incubate your primary antibody solution with that membrane. After that is done, I mean, you can incubate it for 1 hour at room temperature or overnight. And once this is done, you pour the antibody solution and use it on a blot which you want to, where you have samples you want to analyze. This purified antibody solution should be used as soon as possible after the cleanup is done. So you have to empirically determine the conditions yourself, because it depends upon your primary antibody. In general, storing primary antibody solutions in milk for longer time can be a little bit risky, because bacterial growth can be induced. Some primary antibodies can be stored frozen in milk after first experiment, after first blot. I know that antibodies are often reused, but not all primary antibodies can be reused. So if you do not get a signal in a second incubation, it means that this primary antibody cannot be reused. It is very antibody specific, and I talked about it in my first presentation. I will paste a link to the recording for you who did not attend my first lecture.
Okay, one more from Miha. Hello, Professor. My second question is, we should check tissue-specific expression patterns first before going for antibody staining. I mean, we should use that specific part which is more abundant in the protein.
Yes, especially if you work with proteins of low expression, then there is a database where you can check expression level. I will paste a link to it. It is also in my first lecture. So is it a protein abundance database? And in that database, you will see protein level for, it is different species in that database. So this is Arabidopsis, maize, human, Drosophila, and also there are other databases where you can see where your target protein is located. This is especially valid for proteins of low abundance, like transcription factors. So you have to really take into consideration to analyze a specific cellular fraction in such case. And if you also use a specific organ, for example, very tip of a root or a specific developmental stage.
Okay, another question. How do we know the problem of the non-specific band caused by SDS?
This is a very good question. Proteins, high molecular weight proteins require more SDS than low molecular weight proteins. And this, if SDS is a reason of a problem, this has to be checked empirically. There's no other way. So by lowering the amount of SDS in your protocol. But at first, I would check other parameters than SDS.
Okay, I see there's an addition question there, how to test the transfer condition by weight.
Yeah, I talked about it in my first lecture about how to check that the transfer is done correctly. So please listen to the part 1 and you will find the answer there. It is a very quick way to check if a transfer is done properly. You can always stain the gel after gel electrophoresis to see if there any bands remain in the gel. If they do, it means the transfer was too short. And you can also include, especially for low molecular weight proteins, you can include a second membrane in a sandwich and then stain it in Ponceau, both membranes, after the Western blot, because if your transfer conditions are too harsh, the protein will be blasted through the membrane and then therefore lack of a signal on the Western blot. So you can catch it on the second membrane. So please listen to the part 1 and you will find a little bit more information and pictures.
Okay, thank you very much. Our session will end today. Thank you for everyone's participating. Thank you.
Thank you
Can
very
leave
much.
the call now. Okay, thank
Yes.
you.